Okay, for this video, we're gonna go over Lab 12, Bioinformatics. So first thing was Sanger sequencing. And this is where you get those electropherograms, those like little color columns. What those are visualizing is a process where DNA is being transcribed using the primer in this little system. And at the ends, there are little fluorescent nucleotides, remember ATCGs, and they light up a certain color. So A's light up like that. Let's call it here. And at each segment length, so at each little thing, you can have different signals. I think this is C. I don't know if these actually are corresponding. G and T. So what happens when you're doing Sanger is that when you get one of the fluorescent signals, the chain stops like making DNA down. And so you know where the length is at. And on each one, you can see like which color fluoresces because each nucleotide has different color. So in this case, this this sequence is just going to be A, C, G, T, and you can just go on and on and on by measuring like the length. It's because as you go longer, you can read those ferrograms. So definitely watch a video on this, like an animation. It'll do a lot better than I can do like visualizing this. And the way you can measure the sizes and the fluorescence is, remember you can do those little ETG. So the larger ones go down, the DNA smaller. And so let's see, I'll copy the one in here. It's like here, here. So the smaller ones start down here. And that means that the smaller ones, remember, they're at the beginning of the chain. See how the chain will make its way like here, like signal, 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 signal. So if you're reading the DNA strand this way, the small ones are where you start. That's why you start all the way down here and you read up. So in this case, we have a sequence that goes C, T, G, G, T, A, C. And that's how the computers, and that's how we had to do this. We had to read these sequences like that for a long time. So what you'll probably see, and how that's visualized best, is those, like these. Let's just do this. Oops, which one am I missing? Yeah, blue. And so if I correspond, I don't remember if these are the same colors. But each one corresponds to the same nucleotide. So this is how you can read it across. It's T. Your assignment would tell you, like, if you were doing, I'm just gonna see, if you were doing, so that's how you read it. And that's those signals that we get each time, because they're corresponding to the spikes in those little signals that we get right over here to each one. So the longer they go, the further down the line they are. Definitely watch an animation on how Sanger works. I'm not gonna be able to draw it very well on this, but essentially remember reading this. This is essentially how you're reading it, just like that. And if you wanna draw it, I don't know if you can draw on the tests, but I mean, I would. Okay, next thing is those sequence alignments. All you're doing with those is that